Awesome. Okay. Well, my name is Samuel Lara, and I welcome everyone to this webinar. Just as a quick reminder, so the video did not have a lot of sound, but it basically said that the Soil Community Platform is a platform where we use it to connect with partners, to share knowledge, to work and collaborate and join across different projects and connect with different researchers working across soil projects. So welcome everyone. Today's webinar will be focusing on soil biodiversity, specifically on meso and macrofauna. And we wish to answer the question of how does soil and fauna contribute to soil health and how we can better monitor it and integrate it into biodiversity assessment frameworks. And for that, I welcome our coordinator, Raul, who will be moderating the session today. So Raul, the floor is yours.
Thank you. Can you hear me?
Yes.
Okay. Well, welcome to our webinar today. We continue with the soil biodiversity webinar lines as we started with the microorganisms 1 month ago. So we continue with the fauna, also a group of organisms very important for soil health, soil functionality, that we need to take into account also in our assessment and soil health plans. How we will show today and how important their assessment and measurement is. So we will start with the different talks, and the audience, you can use the chat for questions. We will have a questions and answers section at the end after all presenters has introduced their topics. So you can directly post on the chat your questions to each of the speakers, or you can wait until the end. So we will give you the time to raise your hand and speak with them and ask them their questions. So it's up to you. I will be gathering all the questions you are writing on the chat, so no problem for that. So we can start so that we have more time for the discussion. We will first start with Christine Van Capelle from the Bioservices project belonging to the Tuning Institute in Germany, talking about soil mesofauna, soil indicators, potential limitations, and land use effects. So, Christine, the floor is yours.
Thanks a lot. So I will try to share my screen again. I hope it will work. So maybe you can give me a feedback whether you can see my presentation.
Yeah, I can see it.
Okay, so then we can start. Thanks a lot for the opening remarks and also for inviting me to take part to this webinar. Today we will start with the soil mesofauna, and the topic of my presentation is soil mesofauna as soil indicators, potential limitations, and land use effects. So when we're talking about the soil mesofauna, the first thing that is important is to know who is exactly involved in this group and why are these organisms important. So the soil mesofauna comprises soil invertebrates of between 0.1 and 2 millimeter diameter that live within the soil or in the litter layer on the soil surface. This size class therefore covers intermediate sized soil animals that range between micro and macrofauna, and typical members are mites, springtails, proturans, diplorans, pauropods, or amphitrids, for instance. Despite their small body size, these organisms are of high relevance with regard to soil fertility and therefore also with regard to soil health or resilience. And in this context, some key aspects play a major role. So first of all, they bridge the gap between the micro and macrofauna, so they represent a kind of important link within the soil food web. Moreover, they exert an ecological impact that is disproportionately large in relation to their small body sizes and biomasses. And finally, they are involved in many soil processes, fulfill different functions in the soil, and hence contribute to the provision of a broad range of different ecosystem services, some of which are listed here. So, of course, depending on the community composition, to different extents, they can contribute to enhance carbon sequestration or nutrient availability, enhance the soil fertility, regulate soil water balance, and so on and so on. And now, given their important role in the provision of all these services, combined with their sensitivity against management or environmental changes, they represent valuable indicators for assessing soil conditions. And in this context, especially mites and colembulans as microarthropods, which completely belong to the soil mesofauna and not just partly, play an important role as they contribute different functional groups and moreover are highly abundant in different kinds of soils and in different ecosystems. And now, in order to make use of their indicator potential, different parameters can be determined for both organism groups, like for instance, in the case of mites, total abundances in combination with the relative shares of different mite orders, which differ functionally due to the use of different food sources, provide valuable insights into soil conditions and into the soil food web. With regard to the colembulans, usually total abundances are as well determined, and in this case, it makes sense to identify the organisms to species level, as colembulan species can markedly differ with regard to their habitat requirements or their food preferences. Then, based on the identified species, we can calculate different diversity indices, like for instance, the Shannon or the Simpson index, and as well, we get information about the species richness. Moreover, depending on or based on certain morphological characteristics, the ecomorphological index, abbreviated as EMI, of the identified organisms or species can be determined, and then based on this value, colembulans species or organisms can be assigned to one out of three different functional groups. In this case, we call them life form types. And here we distinguish between euedaphic, hemiedaphic, and admöbion species, which differ with regard to their vertical stratification in the soil profile and their adaptation level to the life in the soil. So euedaphic species live deeper in the soil, admöbion species on the soil's surface, and hemiedaphic species in the upper soil layer. And now, to determine all these parameters, up to now, usually traditional methods are used, which comprise sampling using such soil core samplers. Typically, we sample up to a soil depth of 20 centimeters. The soil cores are then taken to the lab, where the microarthropods are extracted using a high gradient extractor like such a Kempson device, for instance. Organisms are then transferred to ethanol for preservation and finally taxonomically identified using either a stereo microscope or a microscope and the relevant identification keys. All these traditional methods are highly effective. However, there are also some drawbacks or limitations. So the sampling procedure, for instance, is relatively labor intensive, depending on soil conditions or soil moisture, for instance. Moreover, there is the potential of underestimation of very active surface-dwelling species, which might escape before the samples are taken. Then, with regard to the extraction, this procedure is relatively time-consuming. So one extraction cycle lasts about 10 days, and special equipment is required. And finally, regarding the identification, it is also time-consuming if many species if many individuals are present in the samples, and a certain level of expertise is required. And now, given all these limitations with regard to future studies, the method of using eDNA might represent a promising approach which potentially addresses some of these issues. However, at this moment, I only want to mention this method as a possibility because it will be explained in more detail in a following presentation today. So, and now I would like to show you some first results from the project in which we are involved, and this is the Horizon Europe project Bioservices. Bioservices is an acronym for linking soil biodiversity and ecosystem functions and services in different land uses, from the identification of drivers, pressures, and climate change resilience to their economic valuation. And within this project, a large sampling campaign was conducted in 2024, and during the sampling campaign, campaign, 5 out of a total of 7 land use types of 3 intensities each were sampled in 5 biogeographical regions across Europe as indicated on this map. The aim of the sampling campaign is to understand the role of soil organisms in the provision of ecosystem functions and services in terms of biogeographical region, land use, and soil structure. The sampling procedure was performed according to the Lucas procedure, and during the sampling, different kinds of soil samples were taken so that a lot of different parameters can be determined. And in this context, we also analyzed the soil mesofauna, so colambulans and mites in our experimental sites using traditional methods. And now I would like to show you some first results from this project. In this context, as I already mentioned, traditional methods are relatively time consuming, and against this background, some analyses are still ongoing or pending, and therefore these are really preliminary results. So on this slide, you can see the colambulan abundances shown on the left-hand side and mite abundances on the right-hand side, shown for all 5 regions and the 7 land use types. However, I will not consider land use intensities here today due to the limited time. Tables in the middle indicate the statistical significances of region, land use, and interaction effects. And as you can see from these tables, regional conditions, land use types, and also the interaction between both significantly affected mite and colambulan abundances. Individual numbers were thereby highest in the continental region and decreased through the Alpine, Mediterranean, and Atlantic down to the boreal region. And now let's take a closer look at the land use effects in those regions where the abundances were highest. These results are shown here, and from these figures we can see first of all that microarthropod abundances were highest in semi-natural agricultural and forest sites, so that obviously these land use types offered better habitat conditions for these organisms as compared to the other land use types. If we compare the impacts on both organism groups, then we can see that colambulans and mites are specifically affected by land use type depending on region, as impacts differed in 2 out of the 3 regions. Now let's take a look at the taxonomic diversity. So on this slide, total colambulan species numbers are shown for all 5 regions and for the land use types within the regions. And these results indicate that colambulan species richness significantly differs between regions. So here we found the highest number of 81 species in the continental region and the lowest number of 12 species in the boreal region. With regard to the land use types, we can see that land use effects on species numbers significantly depend on regional conditions. Here we found highest species numbers in agricultural, forest, and urban soils, which are not exactly those land use types in which the abundances were highest. Now, based on the identified species, we calculate different diversity indices, and on this slide, exemplarily, the Shannon index as an alpha diversity metric is shown. This value ranges between 1.5 and 3.5, and most of you are most probably familiar with this ratio. And if we take a look at the results, then we can see that this value ranges between 0 to 1.8, indicating a low to moderate taxonomic diversity in our experimental sites, with decreasing values from the continental through the Alpine via the Atlantic and Mediterranean down to the boreal region. Moreover, we can see that a highest species richness is not necessarily linked to a highest taxonomic diversity, so that one of these parameters does not allow conclusions about the other. And finally, regarding the land use effects, we can again see that impacts differ between regions, and here we detected a highest taxonomic diversity in 3 regions in agricultural soils, in 1 region in semi-natural soils, and in 1 region in urban soils, so that obviously agricultural soils are not that bad with regard to colambulan alpha diversity. Here you can see some findings for the functional diversity. So here relative shares of the ecomorphological index and the life form types of the collembolans are shown. Different colors thereby indicate the different life form types, and these findings reflect, first of all, that the share of oedaphic species is higher in the boreal region than in all other regions. A possible explanation here might be that in this region we have long frost periods, and these organisms are best adapted to such conditions as they can escape by moving down deeper into the soil. In all other regions and land use type combinations, the most common EMI is 0.5, so that obviously here hemiedaphic life forms with an intermediate expression of morphological properties are best adapted to the soil conditions. The most balanced functional distribution is detected in the continental region, and this is also where we found the highest taxonomic diversity and the highest collembolan abundances. Now, regarding land use effects, we again find that land use effects on the functional diversity depend on regional conditions. Here, the highest diversity was detected in agricultural, wetland, and urban soils, whereby agricultural soils again exhibited a comparatively high level of functional diversity. However, if we now compare the land use and region combinations in which taxonomic and functional diversity were highest, then we can see that both parameters are not associated in 3 out of 5 regions. Finally, some short results with regard to the mites. So here you can see the relative shares of mite orders, which indicate the functional group decomposers, predators, omnivores, and fungivores. And these results reflect that the share of fungivores omnivores, is higher in the continental and Mediterranean region than in the other regions. So these organisms are generally considered to be generalists. Nonetheless, they have some positive effects as they can adapt easily to the food supply. Populations with higher amounts of omnivores are therefore less likely to collapse, and so they contribute to ecosystem resilience. In all other regions, decomposers dominate the mite community, so that we can assume here to have an efficient and fast mineralization, leading to nutrient-rich soils which promote plant growth. Finally, with regard to the land use effects, we again found impacts that differ between regional conditions. Here, the highest diversity was detected in agricultural, wetland, forest, and urban sites, whereby the urban soils showed the highest functional diversity in 2 regions. And for me, this is quite surprising because we often assume that urban soils are not that good as habitats for soil animals. So just a short summary. The first results from bioservices indicate that collembolans and mites are widespread and abundant in the alpine, continental, and Mediterranean regions, so that here they have a high soil indicator potential. The results thereby reflect that land use effects on soil microarthropods depend on biogeographical conditions, that impacts differ between collembolans and mites depending on region, and that all analyzed parameters are specifically affected, as again indicated in this table, in which those land use types which exerted the most beneficial effects on all parameters are listed. In the Atlantic region, collembolan abundances and diversities were lower. So here we would recommend to decide on a case-by-case basis whether these organisms are valuable indicators. And in the boreal region, we would recommend the use of alternative indicators, as abundances are very, very low, and therefore here we assume a low indicator potential. And this brings me to the end of my presentation. I thank you a lot for listening, and I'm available for questions if there are any. Thank you.
Thank you very much, Christina, for your presentation. So we will move to the next speakers. Now we have our question and answer section afterwards. So, as said, we move to our next speaker. Sorry, I have to open the file. I think it was closed. I don't know why. Okay, so Stefan Schroeder next, also representing Bioservices from the Turing Institute, the same as Christine. We'll move to macrofauna, just to also add this type of organisms in the webinar. So, Stefan, go ahead.
Yo, thank you so much for inviting me to this webinar. Can you see my presentation?
Not yet. No.
Okay.
Yes, now we can see.
Now we can see it.
Perfect.
You can start, Stefan. Thank you.
So, thank you very much for inviting me. And so I want to share results with you from two EU projects. And I want to talk about management and land use effects on earthworms on a European scale. Why earthworms? So just to give a short overview, they represent important functional groups as chemical engineers, biological regulators, and ecosystem engineers. And as we see in this table, they are involved in a lot of important soil processes and provide important ecosystem services. So that's the background why earthworms are worth to study in such projects. And I want to share results from these two projects, Soil Diver Agro and Bioservices. Both are large projects, and at this place, I want to thank you to all the colleagues who were involved in Soil Diver Agro or are involved in bioservices. And thank you to them for all their effort and all their work to make these projects successful ones. So in both projects, we did earthworm sampling in the same way according to ISO standard. First, you start with an excavation of a soil block, and then the soil block is analyzed during hand sorting for earthworms, and at the same time you can put a mustard oil solution into this pit and wait for deeper burrowing earthworm species emerging, coming out of the soil. And then from both parts, you collect them in a tube. And in the lab, we have a lot of parameters we consider. It's the abundance, biomass, species richness, the age structure of juveniles and adults, and ecological groups. As we know, epigeeks, which are more on the soil surface and in the litter layer with more horizontal burrows, endogeek, species which have a network-like burrow system in the soil profile, and the deep burrowing anesic species with vertical permanent burrows. So first, in Soil Diver Agro, we considered a pedoclimatic gradient, and we were studying two farming systems, two management systems, conventional farming and organic farming, in 9 pedoclimatic zones from Boreal to Mediterranean South. And in each of these zones, we sampled 10 wheat fields per system, and we always took into account that organic and conventional farming fields are more or less close together to have quite comparable soil conditions. So this gradient is reflected here in the temperature, precipitation, and soil organic matter. We see these boxes from right to left. It's from north to south. And according to the mean annual data, over the last 30 years, we see an increasing temperature from north to south and a decreasing soil organic matter from north to south, and an inconsistent precipitation image for rainfall. Okay. In total, we identified 19 earthworm species, and we see here again from left to right, from north to south, that we have different species in the climatic zones. There's one species, Abrictodea rosea, which we found and identified in all these zones, and there are 6 species which were only found in one of these 9 zones. In total, we had 3 epigenetic and 10 endogenetic species, 2 epiendogenetic species, which are intermediate between both ecological rules, and 4 anesic species. Here we see from the top to the bottom the abundance, the biomass, and the richness, species richness in these graphs. Again, left to right, it's from north to south. And we see that we have that the pedoclimatic gradient significantly explains the individual density and the biomass from north to south. And we see that the highest species richness we found in the Nemoral, Atlantic North, and Atlantic Central region, less species richness to north and south of these regions. Continental is a little bit surprising with these low richness, because in the year of sampling, there were so dry conditions already in the spring that this is more an underestimation what we see here. Surprisingly, there was no significant difference in earthworm metrics regarding the farm systems. Here we see the first 9 graphs on the top. It's the abundance and the species richness on the bottom. These 9 graphs each represent the different pedoclimatic zones. And we see that in total, there is no significant difference, but also in each region, we don't see any difference. So the blue boxes are conventional management, the green boxes are organic farming. Okay, we know from literature that in specific experiments and so on, it might happen, but it was surprising for us to find this pattern all over Europe in all regions, and that this result said, okay, now we have to look more into detail into the metadata of the management of both farming systems. And then it was very interesting to see that the difference in management measures were quite low. And so, for example, here we see again in blue, it's conventional farming, green is organic farming, and from top to bottom, we see tillage, fertilization, rotation, residue incorporation, legumes, and pesticides. And for example, when we start with tillage, we see that reduced tillage was quite common in conventional farming. 25% of all farms in conventional farming practice reduced tillage, which was not the case in organic farming. We know organic farmers in most cases need to plow to combat weeds in their fields. And we also see that we have also crop diversification in conventional farming. We have longer rotation cycles. We have residue management also in conventional farming. Farmers started to consider legumes in their rotation. And finally, we also identified that the pesticide application was okay, it was there in the conventional farming, but on a quite low level, which obviously was not harmful for the earthworms. And that is the main message, that we see that obviously an increasing part of farmers now picks up management measures from organic farming to be more sustainable in their management, and which means at the end that we do not have any longer these two distinct boxes with conventional farming and organic farming, so that it merges more and more in the future, we assume. Okay, we move now to the other project, to bioservices, which Christina already introduced. And so, yes, 5 biogeographical regions with 7 land use types and 3 intensity of management in each land use type. And these results are still preliminary, what I present, because the statistical evaluation is still ongoing. What we see here is just an overview over the 5 regions, which with the different land use types. The colors: brown is agriculture, forest is green, industrial, black urban, orange, yellow semi-natural, gray is mining, and blue wetland. And we see that we have the highest abundances. So what we see here is abundance individuals per square meter in the Alpine region and in the continental region. So very low numbers and only 2 species in the Mediterranean region. And in each region we have land use types which are quite high with the abundances. So in the Alpine region, it's agriculture and semi-natural. What we also saw for the microarthropods in Christina's presentation. In Atlantic region is in urban and wetland. Boreal region, agriculture and wetland. Continental, urban and semi-natural. So agriculture and semi-natural is has very high abundances. And so in total, we have 20 different species, 9 epigeic, 7 endogeic, and 4 anesic ones. And we see that we have highest numbers in abundance and also highest biomass in the continental and alpine region, followed by boreal, Atlantic, and finally the Mediterranean region, and that we have highest abundances and highest biomass in agricultural and semi-natural land uses, followed by urban, wetland, mining, forest, and industrial. So we have a concentration here in the Alpine and continental region, and to the north and the south it is decreasing. So in the following, we focus on the two regions, Alpine and continental, and now we have a detailed look to the three different intensities. One is high intensity, three is low intensity, and two medium. What we would expect is what we see here in mining for continental, that with increasing intensity, the abundances, which is presented here in the graphs, decreases. But interestingly, we also see that in agriculture, for example, we found an increase in the high intensity after a decrease with medium management intensity, or the other way around, in medium intensity abundances and also the biomass increases and a little bit decreases with high intensity, but is still above the low intensity. And in semi-natural, you can even find that with increasing intensity, the abundances and the biomass of earthworms increases. That means that, of course, management measures in land use systems can support earthworm communities, and it depends on these management measures. For example, here in semi-natural, it depends what we considered in the intensity are different frequencies of mowing over the year. Or when we see wetland, okay, more intensity in the land use means more drainage, which is better for soil fauna in total, because you have the soil is not saturated with water, and you do not have reduced conditions and so on. So the conditions are better. Or when you have in agriculture, for example, more applications of organic matter, of residues, and so on. And you have diversification. Then also you can have a higher intensity in the management, but it can be more sustainable and supporting for organisms. And that means intensity of land use can mean both positive or negative impact and depends on the management measures. Finally, to sum up what I have presented, we saw that the pan-European gradient of pedoclimatic conditions forms earthworm metrics, and that is an important message, that conventional farming increasingly converges with organic farming by adopting sustainable measures from organic farming, which blurs the management differences between both systems. And this is reflected in our case in the lacking differences in earthworm metrics. And in bioservices, in relation to the studied land use types, agricultural and semi-natural are most convenient for earthworms, and industrial is most detrimental. And finally, depending on management measure, the intensity of land use causes negative impact on earthworms or can even promote them. Okay, that is what I wanted to present. And thank you very much for your interest and for listening. Thank you.
Thank you very much, Stefan. So, as said before, you can post your questions on the chat or wait until we have our last session and you can directly ask the question you have. So we move to our next speaker. Now we move to we've been showing results from bioservices project. So now we have University of Vigo, the coordinator of our sister project Software is here, represented by Julia Cunningham. You are here, so you are gonna speak about the comparison between molecular morphological approaches.
Hello everyone, I hope you can hear and see me well.
Yes, we can see your screen, but not the presentation.
Perfect. Give me a second. It's now there?
Yes.
Perfect. Okay, let's
Perfect. Thank you.
As we heard before that there were several morphological approaches used to
sorry, I don't know if it is me, but now I stopped seeing the presentation.
Okay.
I don't know the others. No, now I see it.
And if I go to full screen, it's not there.
Now it's like Zoom benden des Molvidumodus Klichen.
Okay, if not, we stay like this. I don't know. So like this, you don't see it?
No, it's again Zoom benden des.
Okay. I can try with the PDF otherwise.
Okay.
Give me a second if we have one.
I don't know if you can better approach a microphone because on the chat people are saying that the voice is low. I don't know if you can speak closer to the microphone or...
Okay, or I can speak louder.
Okay.
We're good now?
Okay, I hear you, but some of them they can say, and I see the screen now. Still low saying, but I hear you, but
Okay.
but low. Maybe you have to speak louder.
Okay. I tried that, and if not, please let me know. I will
Okay.
then continue. So yeah, as you said already, that I'm part of the SOC4S project, the partner project of bioservices that we saw now presented by other colleagues, and I'm working at the University of Vigo. And I want to present you today different methods of assessing soil biodiversity, because before we've seen several morphological methods that are based on visual characteristics of the fauna. But here I also want to add another method that is more and more used, that is molecular methods, and especially using soil, environmental DNA or eDNA short, using either DNA metabarcoding or DNA metagenome analysis to assess the soil life. And also community DNA, which means that the tissue of the soil fauna is sampled, is used, and this is gaining importance. And here I want to start with a European-wide study, which is called Lucas Soil, that started in 2018 with a first time.
Sorry, Julia, I don't know if you're moving the slides or still in the first one.
Okay, yeah, I'm moving.
Because we don't see moving them.
Okay.
I'm still on the title slide.
Okay, I don't know. Technique is not on my side today. I hope you see this light here now, even small, maybe, or still not.
I only see the title.
Okay. Otherwise, maybe you also have the presentation. Otherwise, maybe you share it.
Or maybe Samuel, maybe you can share it. What do you think, Samuel?
Let me, I just found a problem. I think I was in the wrong.
Right. I have a copy here as well if you wish, Juliana, that we can share.
It's moving now?
Yes, now we see it moving.
Perfect. Okay. Sorry for this. So I was presenting you the two different methods available with the morphological and the molecular, and I started then with the Lucas Soil Survey, the European-wide assessment of DNA, which was firstly taking place in 2018 with 1,000 sites. Then it was repeated in 2022 with already 2,000 sites. And here we also took different sample sizes of DNA, not only the 0.2 grams that we're taking 3 times, but also 3 times 100 gram in some test sites. And then again in 2027, next year, this will be repeated, but it's still not known on how many sites. And here assessed are not only microorganisms with archaea, bacteria, and fungi, but also other eukaryotes with the 18S gene assessed. And just a little insight into the workflow, and that here we take 5 samples from a central point and then in northeast southwest direction, and that all these sample processes are online available. Important for you in case you have further questions also, and then also that it's you can see the green square around the soil biodiversity processing, and some results and publications that were so far published on one side on the microorganism, but also then the eukaryotic diversity, which was a publication in 2023 where we assessed 787 sampling points in the 6 different vegetation types. And here we not only had croplands, managed and unmanaged grasslands, and also different types of woodlands that we compared. The samples were taken in different seasons, as you can see, and throughout Europe. In the different, here on the left on this map, you can see the different ecosystem types, and on the right, then the different seasons when the samples were taken. I just want shortly to mention the methods behind it, because we have still methods evolving, which I think is still important to point out. Some decisions taken, which extraction kit was used, which platform then to sequence, and also that we took ASVs, so which means 100% OTU similarity, which taxonomic assignment was used here with Silver database, and that we also took into account environmental data, but also apart from the abiotic factors, also other biotic factors like the respiration quotient, basal respiration, or microbial biomass carbon. Some of the results that we found here are that higher land use intensity, here presented on the left by this land use bar, with agriculture and annual agriculture in yellow, in orange with permanent agriculture, then with the 2 different grassland types in green, with higher intensity on the left and lower on the right, and then the 2 woodlands on the right side that you can see with higher intensity, the diversity was decreasing. And this was not only the case for fungi, but also for protists, and then also for the soil fauna, here represented by rotifers, tardigrades, nematodes, arthropods, and annelids. So there was a similar trend with some variation, but this was an important finding that we had there. And then also when we looked at the overlaps between the 2 different 3 groups that we had with the croplands, grassland, and woodlands, we found the highest overlap between croplands and the other 2 ecosystem types, which means also that it can be a hint towards that croplands represent also the legacy effect of being formerly a grassland or woodland, and that those DNA strains are still remaining in the soil. When we compared the beta diversity between the different ecosystem types, it was clearly to see that it differs between the different ecosystems as we expected. So here yellow again the agriculture, green grasslands, and red the woodlands, and also then which variables were driving the different diversities and between those ecosystem types. And then looking at the homogeneity and of the multivariate dispersion, we found the highest homogeneity in agriculture. Here, a bit difficult to see, but it's the yellow bars on the left that were always the lowest, especially here for the ecosystem type. So this was an interesting finding that croplands seem to be more homogeneous in comparison to other ecosystem types. An important conclusion here was that different drivers were driving the biodiversity in different ecosystem types, but that the highest diversity was found in croplands, but also the highest homogenization, and that there are important factors still missing, which was, for example, the volume taken, because we are taking a very small amount, and it might be that it doesn't capture well the bigger soil organisms, especially the annelids data was very suspicious. Then also we don't know enough about the necromass and the legacy effects of DNA that is no longer of alive organisms but remains in the soil by like necromass. And then also we don't have enough information on other land use practices. So here is still some future research needed, which is then while we compared findings of the Lucas with other European-wide studies, Ecofinders and Soil Service, that were looking at morphological methods across ecosystem types. And here we find quite some variation. So you can see that Lucas in green here, having these higher diversities towards higher intensely used ecosystems, towards croplands, while the opposite effect was found for soil services and Ecofinders. This brings me now to the SOP4S results, which is this European-wide study that we are doing at the moment. Here you can see on the map the different sites taken. It's a spatial sampling across 430 sites in 9 pedoclimatic zones, but also a temporal sampling involved with 5 different land use types and 5 time points. And also here you can see the different ecosystem types taken, ranging also here from arable forest grasslands, orchards, but also urban and wetland in addition. And the sampling design is captured also in a protocol that is available, but also here visible in this 3-plot design where the macrofauna is taken as a monolith, and then also for the microarthropods, a 10-centimeter soil core that is then helping to excavate, while the DNA is taken from the auge, which is here presented as the soil sample. So it's like 4 points around the monolith core, and also from 10 centimeter. And here we are taking in for some organism groups as well the community DNA, as well the eDNA represented by the 18S gene, but also then the hand sorting represented here by this microscope on the side of the extraction. Here just a short introduction into the eDNA primers and also the community DNA primers that we are using here for further processing, but I won't go into detail here. And then some findings here for the 18S DNA result. We found some big but significant compositional shifts and also heterogeneity differences between the grasslands, for example, versus the arable land use. And also here you can see with the Hellinger PCA that here we found variation between the different ecosystem types used as expected. And for the beta the dispersion, here it seems to be a higher dispersion between the arable and the grassland as to be significant. And then also looking at different the different climatic zone, as we also saw earlier with Atlantic compared to continental and boreal, for example, there are also variation within those climatic regions that need to be looked into closer, and that also significant indicator taxa by land use are depending a little bit on the organism group we are looking at, and then into the different ecosystem types as well. While we are looking at the macrofauna, we are also comparing the different intensity levels and found, for example, in red represented here the higher intensely used land uses to be in often cases lower compared to the less intensely used management practices. And this was not always the case. Also for example, for urban, we found higher diversity in the higher land use intensity, but here also it's a little bit depending on we need to look closer also then of the impact, for example, of climate, and then how these intensity uses are actually defined within those groups. So here we still have some more analysis to do. So these are some preliminary results. Also for core human community analysis for the land use types, it was visible to see that different groups seem to be different in differing with different ecosystem types. And I also want to show you some of the photos of the macrofauna that we are analyzing and this diversity that we are finding here within the project all over the continent. And that so far it seems the molecular methods, they are allowing a larger scale assessment, also for sure the assessment of microfauna, and it seems to be less resource intensive, and still that there are some shortcomings, as I mentioned before, while the morphological methods seem to be more accurate, that also image identification, automated identification can help here, but it's still more resource intensive with the taxonomists that are often missing, and it also depends a lot on the expertise of the person. So for the implications and the take-home messages, it is still important which methods we are using, and that the land use impacts on soil biodiversity depend strongly on these methods that we're using, and that the molecular methods with the eDNA is powerful, but it's still not perfect and still evolving, and that the morphological assessments are very essential still, and we further need to integrate both methods and to see how those results can be interpreted also in a way that they are actually can be used for policy making, and that we also need more standardization and validation as a critical aspect for the soil monitoring law also in future biodiversity targets. That's it from my side. I thank you very much for your patience, also for the technical issues for solving them, and I'm looking forward to your questions later on.
Thank you very much, Julia. After the initial problems, we could follow you. So we move to our next speaker. We go to another project, so RISE. It is going to be presented the main results, also now introducing the stakeholder engagement with soil health assessment, and in this case with soil fauna assessment. We presented by Pia Oitinowa from Boku University. So, Pia.
Yes, hello.
Yeah. Hey, yes, Pia, are you here?
Yeah.
Okay.
Or you can hear me, yeah? Okay.
Yes, now we can see not presentation mode so far.
Not? Because I've in the presentation, okay. Interesting.
So we saw the PowerPoint, but not the presentation.
Not the presentation mode, yeah. The PowerPoint was there.
Then maybe like this. Can you see it now?
Yes, we can see it right now. Perfect.
Good, perfect. So thank you very much for inviting me. And I like to talk about how to engage farmers. Just a second. How to engage farmers in soil monitoring via citizen science. So our project is called Soil Rise, and I will mainly present you my view for my Austrian view, because the difference between the countries. What are our aims? Of course, data availability, that we want to find a lot of data or get a lot of data, and then also the awareness, so that farmers get more aware of earthworms and their ecological services, as Stefan already told about us. And I think it worked, so in the past and even now a little bit better. So what have we done? This is a biodiversity project in Austria, as I already said, but also in Ireland, Poland, France, and in Germany. And we were thinking, how can we best approach farmers? Because this is maybe not the easiest audience, yeah? Because time is money and earthworms are not tractors. At least when I'm working with students and there is a tractor driving in the background, everybody turns his or her head, but they're not listening to me anymore. And actually these are young farmers, aren't they? So we were thinking of, or we thought about a mentor-based network. That means we have this project team, we have our PhD students, and then we have mentors, which are, in my case, mainly bachelor students and master students. And they want to do their thesis with me. And therefore, I asked them to find 5 citizen scientists who are interested in earthworms and who wants to sample with them. So for the bachelor students, just 5. And for the master students, I've asked to find 10 different citizen scientists. We also have volunteers. These are agricultural advisors, these are governmental employees, or just interested people. And you can see our map from Austria. We have we started 2024 in autumn, and our last season, next autumn is our last season. We've already have a fine network of mentors and also citizen scientists. And how is this working? So we have the mentors, and they usually have family, friends, and neighbors. And what we want them to do, please ask your family, ask your friends. You're coming from a farming background, please ask them if they want to participate. And who can decline if a niece is asking or a nephew is asking, can we sample earthworms? Yeah. So our rate is almost 100% because nobody can decline if a relative is asking. Yeah. So volunteer, yes, but not so volunteeringly. Yeah. So there's a little bit of pressure on the farmer side, but it works very nice. We've sampled a lot of sites already. So this data, what I'm showing you, or the sites that I'm showing you, is from 4 seasons. So as I said, 24 in autumn we started, and then we have 25, actually 3 seasons, because the last season is not yet included. I mean, Austria is not very big. We just have 500 kilometers from the east to the west, but we managed to sample in all governmental countries. We have from sea level, we are from 114 meters up to 1,780 meters. And similar for the precipitation, so for 472 to 1,872 millimeters. So we have almost everything included. As you can see in the map, agricultural land is mainly at the border of Austria and in the east, and then in the middle of Austria we have grassland, and then we have a few gardens here and there, even in the far west. Yeah. So far we have about 286 sample sites. That includes everything, so from farmland, garden, and grassland, and urban lawns. So we also were able to manage or to sample in Vienna in some parks, and found about 5,000 adult earthworms that we were able to identify. And how did we advertise our work? It is, on one point, it's very, very simple, but maybe if you're not in the community, it can be difficult. But how to get into the community is visiting farmer field days. So we did this quite a lot. The recent one was soil evolution 2 weeks ago. That was in Switzerland. Soil evolution is a field day for German-speaking farmers. That means Switzerland, Germany, and Austria. And this year it was in Switzerland. The next time it will be in Germany again. So we offer workshops there. We offer workshops there for students, also for farmers. And usually they are very, very high demanded. So we have always a group of 20 to 50 people and offer up to 4 workshops per day, yeah, or even more sometimes. And we get some very, very interesting questions and ideas and discussion with the farmers. We also offer other types of workshops for farmers mainly, or talks, webinars, online and in person. And if then our mentors are coming and ask if this farmer is interested in earthworms, they usually have heard about us and they agree to participate. That's also how we get contact to our mentors and our, not only as students, but also the other mentors. So they learned about us and they found our project and initiative very nice. And yeah, that's how we get all these citizen scientists. And yeah, that's great. It takes a lot of effort. We've trained so far in this workshop about 9,000 participants, which is a lot. But I think that is also part of the success why we got so much data within such a short time. Also, my students usually love to work with farmers and like to participate in the project. And I think, the project will end next year, but I think I could continue this forever. Yeah, if I have the resources to identify the earthworms, because this is not done by the citizen scientists. Yeah, we do this in the lab. We have trained experts, which I'm very, very grateful because these are very, very good students. And yeah, so if I could keep them forever, I could do soil rise forever because it's working very nice. Luckily, I have something to compare. So my colleagues from zoology did in the same time monitoring across Austria. So it was very, very well identified sites where they took the samples, 412 across Austria. Nearly the same as we did with arable land, with grassland, field margins. They published a fine paper about it. And I just compare our work against their work and see what happened. In terms of numbers of species, in arable land, we found 3 or 4 species more than with the systematic monitoring. Grassland, they were a little bit better than we, but they've also had these alpine meadows, which we hadn't. Then field margin and lawns are the same. We have in both projects about 4 species, which we are not sharing. So they have 4 different species than we have. And we have the same number of species that we share. And in total, actually, for arable land, for grassland, and for this field margin stroke lawn. If we come to the overall total results, we have 1 or 2 species that we found in soil rise, which they haven't found in this systematic approach. And so that means we have also in total a little bit more species than they have. And if I add this orange box on top, a total, if I add this season, then we have now 8 species which we have and which they haven't found in the monitoring. And my conclusion is that you should, if you're not yet worked with citizen science, you should not be afraid. It's actually quite fun if you like to talk to farmers. And why have we found more species than they have, even with not so many sites as they had? Because we were able to also sample in gardens, so which they had no access. So that's my conclusion. Why we were just lucky. And yeah, you know, the sites were not selected. We haven't declined any citizen scientists. They were completely random. They were completely free to search wherever they want to search for earthworms. The only restriction was, if you have decided to sample a field, stay within the field, don't sample across, I don't know, a meadow, a field, and in your orchard or something else. Yeah. So that was the only restriction which they had. And yeah, it worked very well. And yeah, also working with farmers, I always say they can smell if you can drive a tractor. That usually helps. So I can drive a tractor. Maybe that's why we are quite successful in Austria. But actually, I see that all farmers in Austria are now more open to talk with scientists and are open for research, understand results very well, and try to implement them into their daily work. And that makes it even more fun talking with them, because they have some very, very nice ideas and very, very nice, do very, very nice things. And support earthworms. And I think that's from my side everything. Thank you very much.
Thank you very much, Pia, for your interest in us. Yeah, for me, surprising result with so many people engaged. Thank you and congratulations. And so we move to our last speaker, Jérôme Cortet. Sorry, here.
Hi, can you hear me?
Yes, from INRAE. Yes. Yes, we can hear you.
I will try to share my presentation.
Maybe Pia has to stop sharing her screen.
Because Pia is still sharing. Okay. Pia, can you stop sharing so he can?
Can you see it?
Okay. We can see it. Yeah, not on presentation mode.
Okay. And like this?
Yes.
Okay. Thanks a lot for inviting us to giving us the opportunity to present this project, which has been. I'm only the speaker, and there are many people working on this project behind. So this project is in fact began in fact some years ago with this picture you can see here. The project is called Réseau de mesure de qualité des sols. So this project began in 2000, and you can see on this picture that it consists in fact on a regular grid in France, and each 16 kilometers you have a site, and on the site, on each site, you have some indicators that are measured. So each site is sampled every 10-15 years. We are now at the second campaign, because of course we cannot sample all the sites each in 1 year. I have to say also that it concerns also overseas territories. Many results have been published concerning this program, RMQS. Here, for instance, you can see a study on pesticides in soils and also on organic carbon, but this is not the topic of today. We also, from the beginning, at least from 2005, began to work on soil microbes. And so we were able to publish, and especially the team of Dijon, of Lionel Ranjard, and they published an atlas of mushrooms and bacteria, and some publications also on microbes. However, as you know, there are many other taxa in soils, and until now, these taxa were not involved in the indicators that were assessed in this project. So in fact, we built a new project, which is called RMQS Biodiversity, which is so an extension of the RMQS project. And there are many teams working on this project in France, which is led by the INRA, and especially in Orleans in France. And here are the people behind the project, because we are people and not only labs. And so, yeah, we reused, in fact, some of the sites that are in the RMQS program. And for the moment, we have chosen around 300 sites to be sampled until 27. And we have 7 land use types. And you can see on the map that we try to cover all the country. Only the hexagonal part of France now. You can see on the screen that we have 3 crews, east, west, and south. This is just for organization of sampling, because we have three big teams that are organizing the sampling for each year, one in the south, west, and east. So we sample about 70 sites per year. And we do that usually in spring, because we think these are the best conditions for soil fauna. So here is the sampling design at each site. So in blue, you can see the initial RMQS sampling design. And we added so the biodiversity part besides this. And we collect corals. We have pitfall traps, hand sorting, and about the different methodologies we sampled. So we sampled for microorganisms, nematodes. Of course, we have the soil properties. And we tried also to sample for eDNA. So it's about 25 samples for subsamples for each site. We sample mesofauna with classical methodologies using corals at the surface, and also macrofauna using 2 methodologies, one specifically for earthworms, using monoliths and mustard, and pitfall traps for macrofauna on the topsoil. So we have decided to let these traps for 28 days. The idea was to catch the maximum of soil fauna during the period. So here you have a table which summarizes the different methods we are using on this project. On the last lines of the table, you have what has been done before and which is already still done. But you can see on the top of the table all the parameters that have been added since 2024. So the idea is to complete, to have an overview of most of more indicators on soil biodiversity. We try to publish the first results of the first campaign of 2024. So it concerns 69 sites. So we did the classical analysis to see if we attain the plateau for all the groups. If we could also have some relationships with land uses or with climate parameters or soil properties and things like that. So everything is on the paper. But today, that you can see here, but today I will also say that this project is an occasion for us to think about the methods we use. Here you have an example here about carabids that we trap in the in our systems. Here, in fact, we compared the carabids in the different kind of traps. So if we have only 2 traps, or 4 traps, 6 traps, 8 traps, 10 traps, and we tested also a multi-trap directional directional trap. And you can see that in the graph on the right, that if you use more traps, of course, you catch more, you detect more species. So the idea here is not to say that you must use 10 traps, but the idea is to say, depending of the purpose of your study, you might have some compromise. You might have to do some compromises between the sampling effort and what you really catch in your sites. So we try, yes, to discuss this kind of things. This sampling is also a good occasion to compare our data sets and our reference data sets. Here, for instance, for Colombola, we have acquired in the last years, we have participated to different projects. And so we have different kind of data sets. You can see in blue, the blue points on the map are the RMQS biodiv points. But we had also in the beginning of the century worked on the regular grid for the Brittany region in green. We have also, which was yes, a regular grid sampling method. But we had also some projects in other regions, like in red, in the surrounding of Dijon with 300 sites, or in yellow, in Champagne region with about 100 sites. And the idea is to say, okay, if we try to score the different sites we have for Colombola, for species richness, or for density, if you use the different reference data sets for compare your data. In fact, if you try to replace some new sites, like for instance for the project bioindicator 2, you might obtain different scores for the same sites. So this project also is a good occasion to see if we have to be careful with these reference data sets. Another example is about nematodes. Here is about the question of the eDNA and other complementary approaches. And here we also compare our 69 sites of the first year of study. And we have here, you can see on the right, that we have 3 in fact, type of indicators for nematodes: the MOTU, so eDNA, an index based on the species of our groups that are classically identified, and also respiration. And so you have 3 different kinds of indicators. And if we replace the different sites in 3 in 3 categories, in fact, from low to high um, um, effect or high identification, you can see that in fact you don't position the indicators in the same categories. So it means that um, we have for the different indicators, the different indicators in fact are very complementary. So that's our conclusion, and all these conclusions can also be put in the debate on indicators, and that's what we've done also. And currently, there's a strong debate about using eDNA in the soil monitoring law. So we have tried to say that maybe that would be good that if we could use also some of our indicators complementary to the eDNA, and mostly think about the purposes we have regarding the indicator we use. Thanks a lot for your attention.
Thank you very much, Jérôme. So, well, we are we have run out of time, but I will take
Thanks.
15 extra minutes. No, but to start with the questions. Okay. So otherwise we don't have time for that. So we'll start with some of the questions that have been written on the chat. So we can start with Christine. For example, one of them from Jérôme. I am a little bit surprised with your research on Collembola, which are not really consistent with the literature, with low abundance and richness in boreal regions and high diversity in agricultural land use. Would you have some explanation?
Yes, so that's a quite good question. With regard to the arable fields, where we find a high alpha diversity. This is a result which we also find for other organism groups. So in the bioservices project, for instance, protists show similar patterns. There we also find a high alpha diversity in arable fields. Maybe one reason is that here we have a high diversity of different generalist species that are well adapted to the changing conditions in arable fields. However, with regard to the protists, when they compare the results to beta diversity, then they can see that beta diversity is low. So with regard to my results from the bioservices project up to now, I did not compare the community compositions between the different land use intensities and land use types regarding beta diversity. So this is something still pending, but I expect to find similar results here, so that we have high alpha diversities with a broad range of organisms which are generalists and therefore good adapted to changing conditions, but we do not have many specialists and we have a low beta diversity. So this is one point with regard to agricultural fields. Considering the boreal region, I'm not right sure. So here I think that the long frost periods play a major role, why we find lower abundances and diversity. So as I expect correctly, I'm not sure whether here are some colleagues from bioservices from the boreal region. I think I remember that soils have been also waterlogged for a longer period, and this might also play a role in this context, why we find here lower numbers than in the other regions.
Yeah, so they are organic soils and peatlands. So
Yes.
Yeah. Okay, so another for you, Christine, about the Sebastián Echeverría. Why is the EMI only assigned to collembola mites and not to the other arthropods to properly calculate the key BSR index?
Yes, that's also a very good question. So, of course, it would have been nice to have all other soil arthropods as well. These were practical considerations, I think, at the beginning of the project, because we have the situation that our experimental sites are often far away from the institute, so that we needed the possibility to store our samples, for instance, in cool boxes for several days. And this would not have been possible for all soil arthropods to be included in the QBS AR index in the same way. And this was one reason why we focused on these two groups, on mites and collembolans, which are finally included in the project. Moreover, we considered other soil organisms like microorganisms, protists, fungi, and so on. But with regard to the soil fauna, we focused on earthworms, collembolans, and mites for practical reasons.
Okay, thank you. So, another question for Stefans, rather, Sigbert Uvar. Regarding earthworm abundance, how many samples per management intensity and region have been taken for producing these box plots?
Well, that was the same method Christine introduced. So we had 10 Lucas crosses in each intensity of each land use. That means each field with one intensity was sampled 10 times. So
Okay,
it was a
10.
replication of 10.
10. Yeah, okay. Another question for you, Stefan, by Jérôme Cortel. If I understood well, you only found 2 earthworm species in Mediterranean systems. Would you know why? Because we are supposed to have more species in South Europe compared to North Europe. But I might have missed something. Well, in fact, sorry, but I never find earthworms or rarely find earthworms in Mediterranean Spain because of the aridity of the region. But okay, Stefan.
Yeah. Oh, you already gave the answer, because it was the same in the Soil Diver Agro project. It was so dry and the conditions were so bad for earthworms. So as you remember, in Soil Diver Agro, we repeated the sampling 1 year later and so on. And yeah, it depended on the bad soil conditions that we had so low numbers in the abundance, and yeah, it resulted in 2 species. But it's right in other regions in southern Europe, it can give a separate picture. Yeah, but in southern Spain, no chance.
No, it's that way. We are saying that we have to replace earthworms by other faunistic indicator in dry areas.
You have ants.
They move deeper and
yeah.
we cannot find them.
Do you have termites?
Yeah.
Do you have termites?
No, we have
no.
ants.
Yeah, ants. So ants replaces then earthworms and other arthropods, which are adapted to the conditions.
Okay, so thanks. So for Julia, do you think that current databases libraries are good enough for the eDNA to be reliable for a general routine monitoring?
Thanks for the question. I think here a lot of evolvement is needed in the future. That especially for more soil fauna, the bigger fauna we're talking about, the more reliable the reference bases get, and they're not complete yet. So I have serious doubts that they are enough to just rely on molecular methods for monitoring in the future.
Mm-hmm. Okay, thanks. And for Pia, I think that's something that you already answered through the chat, but in case people didn't read, Ivan Gusaropos said, what GIS or GIS app are you using for citizen scientists? Pia?
Yeah, so again, every country is doing this a little bit differently, and we just used the website to make it very easy for the farmers. So the students or the mentors go there, help them sampling, bring everything what they need, for example, the pots with the alcohols for the earthworms, and help them with the data upload. So they actually don't have to do anything by themselves, because they can be very reluctant doing this, and they have a lot of questions. As you can imagine, we have this We have a questionnaire about field history, which we are going to sample, and 5 different countries. So we had to make a lot of compromises. For just one example, are you using fertilizer? Which kind of fertilizer? And one answer was seaweed, which of course we never use in Austria because we don't have sea, yeah? But which was important for parts of France and parts of Ireland. So, and to avoid any misunderstandings, the mentors are helping the farmers also to fill out this questionnaire about field history and farm management.
Okay. And Ivan also asked a question, but I don't really know if it is for you, Pia, or for Jerome. Maybe you can tell us, Ivan. It is, what is the average number of people in a sampling team?
Well, it's in general for
everybody.
Yeah, for whoever wants to ask.
Okay, so for everybody.
It has been already answered in the chat.
Okay, so well, you can go ahead, all of you, to indicate the number of people in a sampling team for the result you have shown.
So during our sampling campaign in bioservices in the continental region, we were between 2 and I think 6 persons, but it was challenging sometimes.
Yes, in France, it's 4 or 5 persons per crew. So we had 3 crews. So it means 15 persons, in fact, for each campaign on the field. So yeah, it's a lot of people, in fact. And this is the this is the most important amount of money, in fact, we spend.
Yeah, I don't know. Okay, so 5 people is the average, I think, for place and campaign. Okay, so for Jerome, what is the key to keep the research project so long and alive? Michaelete said that money, but maybe you can provide more info about how to get these funds, or if the government is involved, or what implications and how to get it.
Yeah, we have the chance in France to have a strong community working on soil biodiversity. So, and we work close together for 20 years now. Yeah, we have some. So we are a kind of lobby, in fact, and so we try to exert this lobby with key people, and also we have the chance to have an agency which is called the OFP, and another one which is called the ADEM, who support us and who defend our projects on higher levels. So that's the way it works. And for the moment, we still have money, but for biodiversity, for instance, we don't know what will happen after 27. So we will see. Okay,
thank you. That's impressive. So a question for all from Unse van Boegen. So yeah, in most of the research I have seen today, there is a distinction in large areas across Europe with different paleoclimatic or biogeographic regions, which shows very clear differences in this area. No? So we are talking about scale. Is there a specific reason why this large-scale approach is used? As an earth scientist, I like to think in different sedimentary environments and therefore different soil characteristics. For example, a forest soil can exist of a sandy soil, but also of a more loamy or even clay soil. I can imagine there are differences in abundances in soil, like these soil-specific characteristics. So we foresee differences with different scales. So is the scale important? And if different scale is used for specific region, resource could be different. So maybe this is mostly for the pan-European sampling, explained by Christine, Stefan, and Julia. What about scales, and if the results are scale-based? When talking about regions, or if we go directly to specific region with different soil characteristics or management?
Sure. Well, it depends on the question you want to answer. And so if you want to get an overview and you want to get responses of groups of organisms to specific management measures or whatever, or conditions, then it's important to have a larger scale. But even in this larger scale, you need also your regional or your local focus to get enough information to compare within the region. So if you have a problem you want to study, which is very specific to different conditions and management measures, okay, then a large scale is not important. But when you want to generalize something, and then it's important to have these large scales and to combine all these different spots you have, and to get an overview, and to get the feeling for the different conditions you have in all these regions. I think it depends on the question at the end.
Yes, and the research question. I don't know if someone else or Julia or
Yeah, I can add also that I think those European projects are important contribution for answering some questions, but we also should not miss the importance of more micro scale, that it's nice to compare the different continental characteristics, for example, but also the soils are so heterogeneous that within even a small scale we have high variation, and we shouldn't forget this perspective and importance to also look more in closer detail here.
Okay, thank you. Okay, I don't know if someone else wants to say something about scales or no. Okay, so maybe we need to finish, Samuel. We are. I know there are still questions on the chat, but I mean everything will be shared, so you will have the emails of presenters, so you can directly approach them and contact them by email and ask the information you need. Like for example, Ivan asked for publication for citizen science, so or maybe you know already, and you can post them on the chat with publication on citizen science, or even you can write directly to them. Well, Maria, about the bioservices season, it was late spring, early summer, depending on the region. May, Mediterranean, going to the coldest regions to June. So I think that we have to finish. Thank you very much. And we've seen how important faunistic groups are for soil health. And we have learned about the different types, how they behave, how important the specific context is. And while we don't always have to say the higher this tumbas involves lower diversity, we've seen that sometimes it's different. So is it true that loss of soil biodiversity is the right indicator when comparing at pan-European level with different land uses? So maybe with the same area with time, but not maybe comparing them as we have seen. So the specific conditions are key with new molecular techniques that can help us analyze the fauna, and citizen science being a good strategy tool to increase the outreach of soil health monitoring, and also that Fauna can be successfully integrated in the national monitoring programs that we've seen in France, that we could take also an example of that. So thank you very much. So Samuel, if you want to wrap up.
Thank you, Raul. A huge round of applause to all of the speakers, and also to you, Raul, for moderating the session. I thought it was very interesting. I just want to let everyone know that the session was recorded and it will be available on the same space. If you want to come and take a look at it later, and the presentations will also be available there. So thank you very much. And please, if you're interested in presenting your work, this is the last webinar of the season. We will be back in September, October with more webinars. So thank you very much, everyone, for joining us, and have a good rest of your day.
Thank you. Bye-bye.
Bye.
Thanks.